Thank you Fred for the introduction. Thank you the PRISMAP. I'm very glad to be here to present you the two projects funded by PRISMAP. Indeed, it is a teamwork. I'm here for the presentation. We were very interested in the development of new in vivo models to evaluate new radionuclide for targeted radionuclide, the targeted radionuclide therapy, and we went toward clinical translation. You all know what targeted radionuclide therapy is, so I can move on. We see now in clinical trials for human use, new radionuclides becoming available. We see a lot of excellent results published with actinium-225. You know that we love terbium-161. We have seen, Zainab have seen a very extensive review of clinical trials. So we see the potential of terbium-161. So it is the image in the violet trial and in the clinical trial running in the Switzerland with the somatostatin antagonist LM3. We were expecting the results, and many more therapeutic radionuclides are becoming available. So the PRISMAP we're talking about copper-67, lead-212, and so on. But these new radionuclides are different, considering their half-life, the particle that emits their range. And we had in mind to develop a new, fast, cost-effective translational model to better understand the difference between these therapeutic particles, beta, alpha, and Auger. Indeed, you know that this model, we were very interested in the development of zebrafish embryos to evaluate the efficacy of short-range emitters, and also the development of tumor spheroids for the evaluation of large emitters, of long-range emitters. Today, I will only focus my presentation on zebrafish embryos because the project is now finished. We have not yet finished the spheroids model. We need a radionuclide with a longer range than lutetium and terbium to compare. The equation we had in mind is to compare lutetium-177, terbium-161, and terbium-155, assuming that roughly the decay of terbium-161 can be approximated by the beta minus emission of lutetium-177 plus the Auger emission of terbium-155. It was the assuming equation. As I said, we get two projects funded. The first one on the second call on 22 with my colleague, Dr. Majid Abdel Khatib, for the zebrafish embryos. And more recently, in the first call of 24, to develop the Auger targeted radionuclide therapy through the palladium-rhodium chemistry with my Italian colleague, Lorenzo T. From a user perspective, for the user selection panel, the grant proposal was quite smooth. The hearings were very interesting. We get very relevant questions that increase a lot the quality of the project. So thank you for your time. The next steps were to sign the convention between the production site and us. Again, it was a very smooth process. It went very fast, so we can run all the projects in time. Let's move on the results on the first project, on the zebrafish embryos. This project was not very easy because we faced lots of challenges. The first is to find the right cell lines that grows well in vivo in zebrafish embryo and easy to transfect for a stable fluorescent staining because we follow the fluorescent staining in vivo. So it was the first challenge. We did plenty of tests. We first use the AR42J cells because the somatostatin receptor 2 is highly expressed, and we have access to the Lutathera because we are working also in the university hospital. But these cells is a nightmare to cultivate. It is a nightmare to inject into zebrafish and embryos. So we move on PSMA plus positive. We use terbium PSMA, but we had issue in the transfection of PSMA. We hadn't stable overexpression of the PSMA over time. So we move on neurotensin compounds, but it is an agonist, so maybe it is not very easy to characterize terbium-161. And finally, we choose my favorite target, GRPR, on PC3 cells, and we are working on RM2, radiolabeled with lutetium and terbium. We also worked a lot to find the best injection modalities into zebrafish embryos. We test intracardiac injection, but lots of embryos died. We try 2 injection into the yolk sac, and we try to pre-incubate cells with the radiopharmaceutical and inject the combo into the zebrafish embryos. And it was the method chosen because of the high success rate. We also choose the GRPR with the PC3 model because the expression of GRPR is so high, and the engulfment into zebrafish embryo is high, and it promotes in vivo metastasis, so it is fine for the study. We face one issue, the customs. For the delivery of terbium-155, we get 2 shippings. The first shipping from Medicis, no issues. We get the 4 megabecquerel for the calibration of the devices, but when working with the proton production at PSI, the production worked well, but the person stayed 8 days at the customs, knowing the 5 days half-life. We couldn't make it with the terbium-155. Let's talk about the results. You see here fluorescence imaging of zebrafish embryos. So you have the control here. We inject the zebrafish embryos 2 days post-fertilization with the radiopharmaceuticals, and we did the acquisition of the result 3 days after. This is the limit. Before 5 days, you do not need any license to use zebrafish embryos. After 5 days, you need a license. So we stopped the experiment at 5 days. So you see the control here, fluorescence staining. We worked a lot also on automatic segmentation on the zebrafish embryos. Control, first day, third day, lutetium around the 4 megabecquerel per ml, and the same with the terbium. So you see on the image that the lutetium RM does not provide any significant decrease on the tumor size, while when increasing the volumetric activity of terbium RM2, we get a significant decrease on the tumor size. We were also able to cut zebrafish embryo, very small, and to do imaging of the Auger emission of the terbium-161 here. So this is the distribution of Auger emission within the yolk sac of zebrafish and embryos here. We are working to obtain a better imaging. This model, zebrafish embryo, is very useful because you can get 3 primary outcomes. The first endpoint will be to measure the area of the primary tumor, the tumor injected into the yolk sac. So we did it with lutetium RM2 with no significant effect, even up to 25 megabecquerel per ml. While for the terbium, we see very nice decrease in the area of the primary tumor. And when we compare, you see that the terbium RM2 at the same volumetric activity using the same specific activity of the radiopharmaceutical is better than lutetium-177 in irradiating very small tumors. But this is the primary tumor. It is a quite large one. Next, we were able to see individual metastasis development into zebrafish embryos, and we compare the difference in irradiating almost individual cells provided by lutetium-177 RM2, which is a GRPR antagonist, compared to the terbium 161R-M2, we see that lutetium, as expected, has no significant effect on the decreased number, the decreased number of metastases, while increasing the activity of terbium-R-M2 significantly increase the number of fishes without metastasis. It means that effectively what we expect and what we have, we did the modeling study with the terbium-161. We were expecting that, but we were not able to separate between the Auger conversion to the conversion electron because we can't make it with terbium-155. So the question is still open whether it is the Auger emission or the conversion electron of the terbium that can provide this effect on the almost single cell metastasis, or at least small cells clusters. You can also follow the mortality of your fishes. And we see a decrease in the mortality with the terbium-R-M2, while we had no effect on lutetium-177 on the mortality of fishes. And thanks to the PRISMAP support, we are now able to make it with patients. So this is the ZebraNet study that we had included the first patient a few weeks ago. Let me show you this study. So we get grant funding for a prospective monocentric study taking place in Bordeaux, France, Southwest, red wine. It is a ZebraNet study. We are working with a clinician, with an endocrinologist, which will enroll patients. We will also work with the surgeon. We track the patient, and at the time of the surgery, the surgeon comes. We take the sample. At the same time, the pathologist confirms the diagnosis of NET. We implant it into zebrafish embryos, and after the patient follow the classical follow-up of NET, which is DOTATOC PET, FDG PET. If no mismatch, targeted radionuclide therapy with DOTATATE lutetium-177, 4 cycle, you are 1 year after, and we evaluate the efficacy with RECIST-1 6 months after the last cycle of targeted radionuclide therapy on the CT. And on this study, we will gather all the data, how the zebrafish embryo can be a predictive marker on the efficacy of targeted radionuclide therapy compared to the RECIST-1. This is the main objective of the study. We had applied to a national grant, surprise after, still with a support letter obtained from Thierry. Thank you again. We didn't get the funding last year, even we get a high score, so we applied again to deploy this technology in France in several centers, enrolling 120 patients to demonstrate that we are able in France to clinically implement this method. And this is the first patient we get. It is a pancreatic NET, so it is not yet NET treated with lutetium DOTATATE. It is a patient which is eligible to NETTER-3. The NETTER-3 trial have just started, and this is a kind of a patient. You see 2 lesions on the PET, on the pretherapeutic DOTATATE PET on the pancreas, no mismatch on FDG, and we successfully implanted it to the zebrafish embryo. It is the first image of a NET sample implanted into a zebrafish embryos. And we expect to get the result of this study. The first analysis will take place in 3 years. And for me, it is the opportunity to make a link with the second project we have in 2 slides. We get also a project on palladium-103 because the potential of the Auger targeted radionuclide therapy. Production was done by DTU, process extremely smooth. Thank you. It was a real pleasure to work with you. We fix a date, and right on time, we get the palladium at the lab. It was excellent. We get 2 shippings, 185 megabecquerel, because the French National Authority not allow more yet. And 2 main objectives, developed a chelate able to bind both the mother radionuclide and the daughter radionuclide, because the rhodium-103M account for 45% of the dose deposit by the in vivo generator provided by palladium-103. So we need a chelate able to bind the two. And if we get lucky, we try to make a peptide with a palladium-103, which has never been done before. And the chemistry part was done by Professor Tei in Italy at the University of Piemonte Oriental. So this is the structure of the chelate, AAZ3S, this is the name code. We, in fact, test for chelate. We test all the concentration, volume, temperature, activity for all the three chelate at low and high activity. You can imagine the number of radio labeling that has been done with the first shipping. And we also work with a colleague in France that derived the peptides targeting neurotensin 1, because it is also a favorite target at the lab. And we were very successful in designing a peptide with nice affinity to the NTS1, radiolabeled with palladium-103, with a nice affinity. Uptake is stable over time in cells over 4 days, which is very long for the neurotensin compound. And we move to in vivo, only LC mice, it is biopsy mice. And we compare the biodistribution of uncomplexed palladium-103, only the complexed AAZ3S, palladium-103, and the peptides derived. And we have what we expect, meaning low activity in brain, in heart, liver, quite high in the spleen, because of the expression of the neurotensin compound, and indeed very high expression or at least excretion in the kidneys. For future palladium targeted nuclide therapy, the organ at risk is the kidneys, so we remove the kidneys and make the first image of the Auger emission of palladium-103, and we see an heterogeneous cortical uptake of the compound. And now we are trying to make a 3D modeling of the dose and to see what will be the risk of administrating high activity of palladium-103 with this compound. And the missing link, indeed, as it is an Auger, we would like to inject this one into zebrafish brewers. This would be a perfect illustration of the potential of Auger and the potential of zebrafish and brewers. And thank you, PRISMAP.